So today, guys, we'll be doing two things. It's on Google Class, we have to print it. Yeah. So we'll be doing two things today. First, we're going to go over properties of compounds. Everything in this lesson is going to be crucial for you to be able to produce kind of the end result of our lab that we're doing tomorrow. Okay, so the information that's in this lesson is going to help you to identify the unknown compounds that you'll be given in tomorrow's lab. So what we're going to be talking about here are the physical properties of ionic and molecular compounds, with some compounds in particular. Certain elements have certain things. Okay, that's what we'll be going over. The information in this lesson will help you to identify your unknowns in the lab. Okay, so pay close attention, make little notes, okay, here and there, especially if I say, hey, this one's super important. All right, the second thing we're going to do is do the pre-lab stuff for tomorrow's lab. So we'll have the Chromebooks for that. Okay, you'll have the template out, and you'll be filling in some stuff, and we'll walk through this first lab report kind of together. Okay, I'll really hold your hand through the whole thing so you get the idea of, okay, here's what I need to say, here's what Mr. Coderre is looking for, okay, in the lab report, blah, blah, blah. So this first one is kind of a 50-50 deal, okay, on how we set it up and do it, okay? All right, so for properties of compounds here, we want to learn the properties of compounds, acids, bases, ionic compounds, molecular compounds, and understand how those properties can be used to identify a compound. All right, so all matter has properties, physical properties and chemical properties. For now, we're only concerned with the physical properties. Physical properties are things that I can measure, observe, and determine without really the use of anything else. A chemical property has more to do with how does it behave in the presence of this other thing. Okay, so you don't really know something has chemical properties until you observe them react with something else. All right, so we're only concerned with physical properties today. That would be simple things like color, density, melting point, boiling point, solubility, pH, whether it's an acid or a base, whether it conducts electricity, whether it's magnetic or not. Okay, all of these kinds of things are physical properties. Okay, we're not going to be using all of those in our lab tomorrow, just some of them. Okay, but they will be very helpful for us. Okay, and those properties that we're going to go over can obviously then be used to help us determine if something is molecular or ionic, and then from there, is it this compound or that compound? All right, so let's do an example here. If I'm talking about the physical properties of something we see and use every single day, water. Okay, what would some physical properties of water be? Like basically describe water to me.
It's clear.
Okay, it's clear, so it's colorless. Okay, what else?
It's a liquid.
It's a liquid at room temperature. Okay, or we could probably say melting point, because that's what determines it to be a liquid, okay, is 0 degrees Celsius. What's its boiling point? 100, right? So those would both be physical properties. I should really pick a better color. That color's really hard to see. Okay, what else?
It's pH.
Yeah. Yeah, and what's its pH?
It's right in the middle, isn't it?
Right, it's 7, neutral, okay? All right, does it smell?
No.
No, if it does, don't drink it, okay? Tip, it's an important tip, I'll give you that one for free, okay? So odorless. Okay. Is it magnetic? I've never seen a magnet attract liquid water out of a glass, so we think we can safely say no on that one. Okay. What else? Does it conduct electricity? Tap water does. Distilled water doesn't. Okay. If you have pure water does not conduct electricity. Right? Because it's a molecular compound. It doesn't conduct electricity. okay? But as soon as you add anything to it, then it does. Okay, so tap water is actually a solution. It's got minerals and things like that in it. The minerals are what allow tap water to conduct electricity, but pure water actually doesn't. Okay? Can it dissolve a lot of things? Yeah, definitely. Okay. So these are all kind of physical properties, right? So if you were looking tomorrow in the lab and there was a beaker full of a clear liquid, okay, you wouldn't automatically say that it's water, would you? Because spoiler alert, there are 2 clear liquids, okay? There's actually a third that's a solution and it looks clear, but it's not a pure substance, okay? But there are 2 clear liquids in the lab tomorrow, okay? One of them is probably water and one of them is probably alcohol, okay? Okay, definitely. One of them's water and one of them's alcohol. If I just look at them, they look pretty similar, don't they? Okay, how could I tell them apart? Lab safety in mind.
Oh, the smell.
Yeah, one has a distinct smell. You can't taste. No taste tests in the lab tomorrow. Some of those will go very badly. Okay, one of them, it'll clean you out, but it'll leave you hollow inside. Okay, so no taste tests tomorrow, please. Okay, but you can safely smell them, right? What's the proper technique for smelling something if you're not sure what it is? Do you put your face over and go? No, not unless you want to be flat on your back with a nosebleed. One of them will do that. Okay. How should you do it? I saw you miming it there. Wafting it, right? Okay. Guys, I have made these things like if it's a solution or something, it's uber concentrated. I like stuff to be sure. Okay. So if it's got a smell, you'll know right away. Okay. If you're doing this and you don't smell anything, it doesn't have a smell. Right? You can be sure of that. So yeah, make sure you waft it, okay? Things like that, just lab safety stuff, which we will talk about a little bit later. All right, so we'd be looking at things like that. If I looked at the 2 clear liquids, I could separate alcohol and water by their smell, okay? I could also separate them by a few other properties as well, okay? We're not gonna be testing melting point and boiling point here or anything like that, but if you swish water around, it behaves differently on the glass than alcohol does. Alcohol will kind of cling to the glass versus water will just kind of go around and around. Little things like that. Their behaviors are obviously different. Okay? All right, moving on. So, ionic compounds. We talked about this the other day. Okay? They are made of a metal and a non-metal. Their bond is due to the fact that they trade electrons. The metal gives them up, the non-metal takes them. But that means that ionic bonds are also very weak. So they are very easily broken. Most ionic compounds, most, but not all, okay, are highly soluble in water. That means that they will dissolve easily in water. When they dissolve, the solution that is made will conduct electricity. And the reason for that is, when you put an ionic compound in water, the ionic bond breaks. Okay, so now you've got a negatively charged non-metal floating around, you've got a positively charged metal floating around. Positives and negatives can help to conduct electricity. So an ionic compound will make a solution that conducts electricity. That is probably the most important feature that you will use tomorrow. Okay, because it will unequivocally tell you the difference between a molecular compound and an ionic compound in the lab. Okay, that is the one way that will always separate them. Okay, so they will conduct electricity in solution. I'll tell you where that could be helpful. One of the unknowns is sugar. One of the unknowns is salt. Do they look a lot alike? Do they both dissolve pretty easily in water? Okay, but salt is ionic. Sugar, I gotta get the numbers right here. Something like that. Is that ionic? That's molecular, right? Starts with a non-metal. Okay, will that conduct electricity in solution? It will not. Okay, good way to tell them apart since you're not allowed to taste anything. Okay, obviously tasting them would be a very quick way to tell them apart, but it could also be a quick death if you try the wrong thing. So, we won't be doing that. I think I covered that before. Okay, So, that would be a way to separate the two of those. A lot of your compounds are gonna be like that. There's gonna be some things that are gonna be very much alike by appearance, but you'll have to run a test to separate. Okay? All right, now, ionic compounds typically exist as crystalline solids at room temperature. Okay, so if you've got an unknown material tomorrow and it's a liquid, is it ionic or molecular? It's molecular, because ionic compounds are crystalline solids at room temperature, and we're going to be doing this lab at room temperature. Okay, so if you have an unknown solution and it's a liquid, you know all the possible ionic compounds are out. Okay, it can't be one of those because ionic compounds are salts. Okay, they are crystalline solids at room temperature. Okay, so ionic compounds will conduct electricity when they're in solution, so dissolved in water, they typically dissolve pretty easily in water, okay? And that's because they have those positive and negative ions. So we got a couple of ways already that we can tell a molecular compound from an ionic compound, okay? But something like sugar, for example, okay, it's gonna behave just like salt does right up until we check its conductivity. Okay, now, like we said, ionic compounds are typically going to be crystalline solids, and if you had the right apparatus, just a little crystal viewer, which is kind of a microscope for looking at crystals, okay, you would see that every ionic compound has a different crystalline structure. And if you really know what you're looking for, that's also a physical property, okay? If you knew, what sodium chloride looked like versus what sugar looked like at a, crystal level, you'd be able to tell them apart. Okay? It's another physical property. Not one we're going to use, okay, but it's there. The reason for them conducting electricity, as I said, if you put salt, sodium chloride, and water, it will dissolve. The two parts will separate. You'll have positively charged sodium ions, negatively charged chlorine ions floating around, and as a result, that'll conduct electricity. I'll show you how that works in a little bit because I've got the stuff here to do it. Some ionic compounds are brightly colored, okay? And that has to do with what metal they contain. Some metals have particular colors for their salts, for their crystals. Okay? Sodium-containing salts are white, okay, as are many. But nickel always makes green salts. All right? So if you've got a crystalline solid as an unknown and it's green, and one of your unknowns has nickel acting as the metal, is it a good bet that that's the one it is? It is, okay? So little things like that, right? Simple observable properties, okay, can sometimes help us to determine what something is. All right, so there's a list of these here, okay, on this page here. So many ionic compounds produce solutions that are brightly colored. Copper compounds are blue. That can be anywhere from bright blue to pale blue. Nickel compounds are green, and cobalt-containing compounds are red, pink, borderline orange sometimes, okay? But in this case, the one we have is kind of the borderline orange, okay? So any kind of reddish tint, okay, to them is going to be a cobalt-containing compound. Okay, questions so far, guys? Now, molecular compounds, they work differently. Okay, as we said, ionic compounds are two charged particles that are bonded together until they decide they don't want to be together anymore. Molecular compounds are sharing their electrons so they stay together. There are no charged units. The lack of charged units means they're not typically as soluble. Okay? And they will not conduct electricity when dissolved in water. Okay. Now, molecular compounds have low melting and boiling points, which means they can exist as solids, liquids, or gases at room temperature. Ionic compounds are only going to exist as solids. Okay, so if something is a liquid or a gas, spoiler alert, there's no gases, they're really hard to contain in a beaker. Okay, so none of your unknowns tomorrow will be gases. Okay, but they can be solids, liquids, or gases at room temperature. So example of a solid, okay, or molecular compound, butter. Okay, butter is a molecular compound. Okay, and it's a solid at room temperature, but its melting point is quite low. It turns to a liquid very easily. Okay, water is another one, molecular compound, right? It's at room temperature, it's a liquid, but you only have to heat it to 100 before it'll turn to a gas, or cool it to 0 before it'll turn back to a solid. So lots of things like that. It can be in any state. Carbon dioxide is a gas at room temperature. Okay. And as we said, molecular compounds are not usually as soluble. Okay, some of them can be, but they're not usually as soluble. Okay, and the solutions they form do not conduct electricity, and nor are they brightly colored. Okay, does alcohol dissolve in water? Can you dissolve one liquid in another? Okay, this stuff is, what, I haven't even got the percentage on it. Oh, there we go. 70% isopropyl alcohol. What do you suppose the other 30% is?
Water.
Water. Are they dissolved? One visible part, right? The alcohol's not floating on top of the water like oil does. Okay, oil is a molecular compound. It's not soluble in water, but alcohol is. You wouldn't be able to mix drinks, not that you guys are old enough to know that, okay, if alcohol didn't dissolve in water. Okay, you pour it in and it just dissolves, okay, it doesn't sit on the top. Sort of make sense? So some molecular compounds are soluble in water, and liquids can be soluble in water as well. Okay? All right, but they will not conduct electricity, okay, when they're in water, because they don't split. Okay, and like we said, molecular compounds which are soluble in water usually produce clear solutions. Okay, hand sanitizer, perfect example. That is a solution of alcohol in water, and it is clear. Okay, if you dissolve sugar in water, same thing, clear solution. Okay, what would this stuff be called?
Hydrogen chloride.
Hydrogen chloride. What's a common name for it?
Oh, no, I was gonna say chlorine.
Right, it has chlorine in it, but no, that's not what its common name would be. It's in your stomach. What kind? Okay, but you're right, this is an acid. This in particular is hydrochloric acid. Okay, hydrochloric acid is actually produced by your stomach cells and helps to digest proteins and other kind of difficult to digest molecules in your stomach. Okay, hydrochloric acid is an acid for one reason. It has hydrogen acting as the metal. Any ionic compound where hydrogen is the metal will be an acid. Are some acids stronger than others? Right? Like they're not all the same strength, right? If I had them all in equal concentration, would some of them be more dangerous than others? Absolutely. Okay? Believe it or not, this is one of the least dangerous ones. Okay? Would that be more or less acidic than hydrochloric acid? Anyone know someone who works in the oil field? Okay? They have to take a course about this stuff. Okay? Their H2S certification. This is a gas that can be a compound that can be associated with drilling. Okay? It can sometimes be released. There can be pockets of it. If you inhale it, it will dissolve your lungs. Okay? It's really, really bad for you. Okay? It's highly acidic. It's more acidic than this because how many hydrogens does it have? 2. Okay? The more hydrogens it's got, the more acidic it's going to be. Okay, this stuff is real bad. Okay? You don't even want the fumes from this stuff to get anywhere near you. Okay? They can cause chemical burns if you're just holding your hands over a container filled with this stuff. Okay? The more hydrogens it has, the more acidic it's going to be. So the more hydrogen ions that would be donated to a solution, the more acidic it would be. Okay? So all acids, all strong acids, are going to have hydrogen acting as the metal. Okay, so acids are compounds that have H plus ions in them, okay, and H is acting, hydrogen's acting as the metal. Okay, that's H plus. Okay, because they're ionic compounds, they are also crystalline solids at room temperature. We cannot acquire them in that form. Okay? We cannot buy them in their crystalline form because they're so dangerous. Okay? We can only buy pre-made solutions of strong acids. Okay? It's actually safer to transport them that way. But they are crystalline solids at room temperature because they are ionic. Okay? Acids taste sour or tart. But of course, we're not going to find that out because, as we said before, we're not going to taste any of them. Okay? But you've had this experience. Lemons are sour. Okay? That's because they contain citric acid, which is a weak acid, okay? But an acid nonetheless, so it tastes sour. Okay? Vinegar is also an acid. Okay? It's an ionic compound. Okay? It's tart. Okay? Borderline sour, but definitely tart. Okay? Vinegar actually looks like this. It's written a little bit differently, okay? But this is the non-metal, and this is the metal. Okay? Because it's organic, it's written a little differently. Okay? But it still has H plus acting as the metal. It's acetic acid. All right, acidic solutions always have a pH of less than 7. Okay? So acids are low in pH, bases are high. Okay, and they will typically turn litmus indicators to acidic, that is red colors. Right? The pH paper that we're going to use in the lab, okay, is just this Hydrion paper. So it's orange at neutral, okay, and then there's a color chart on the side of the container that you use to determine. So you stick the paper in the solution, and then you compare the color on the chart, and it tells you the pH. Okay. All right, so the big thing to remember here is this, okay? They're ionic compounds and hydrogen is acting as the metal. Okay, so if this compound, it's not, but let's say it was, if this compound was in your list of unknowns tomorrow, okay, could you predict what its properties might be? Okay. Is it ionic or molecular? It's ionic. Okay. So it would definitely then be a crystalline solid at room temperature. Right. It would probably be soluble in water, and its solution would probably conduct electricity, and it would definitely have what kind of pH?
Less than 7.
Less than 7. Okay. Because it's an ionic compound with hydrogen acting as the metal. Okay. All right, is that making sense? That's kind of the process that we're going to walk through today and tomorrow when you're looking at your unknowns. All right, bases are the opposite, okay? Bases are also ionic compounds, at least strong bases are, and their solutions will conduct electricity. In fact, you have one strong base and one strong acid as unknowns tomorrow, okay? And they will be your best conductors of electricity. They will be a 10 out of 10 on the conductivity meter. Okay, incidentally, speaking of a conductivity meter, this is what your conductivity meters look like. Okay, they're a 9-volt battery on top of 2 leads, okay, with a kind of face here that'll tell you whether it conducts. Press in the button on the side, the on light comes on to let you know it's working, and you stick it in the solution and it'll tell you how conductive it is. Okay, pretty simple to use. All right, but your strong base and your strong acid will go all the way to 10, all the lights will light up. Okay, so typically bases taste bitter. So if you ever swore as a kid and got your mouth washed out with soap, okay, soap contains lye. Lye is a strong base. That's why soap tastes bitter. Okay, lots of things can be, taste bitter, but they're generally going to be basic or alkaline. Okay, and if they are alkaline, they are going to have hydroxide ions in them. So, as an example, that is an ionic compound that would be a strong base, sodium hydroxide. Okay, that is the active ingredient in oven cleaner. Okay, so it cleans the crud off the inside here, that white foaming stuff. Okay. Which one would be more basic? Sodium hydroxide or calcium hydroxide? Which one has more OH? The calcium hydroxide. Okay, so just like acids, whichever one has the most H is the most acidic. For bases, yeah, the one that has the most OH is the most basic. Okay, so one is, this stuff is sodium hydroxide, this stuff calcium hydroxide, this is lime. Okay, have you ever watched any like of the murder shows on TV? This is the stuff they pour on things to make them degrade more quickly. Okay, it's also something like if you had like a porta potty, okay, it's one of the chemicals they'll put in there to help kill bacteria that make it smelly, okay, stuff like that. Okay, lime breaks down organic material very quickly because it is very, very basic. Okay, a strong base is just as corrosive as a strong acid. Okay, people get this idea that bases are friendly and acids are bad. Bases are not friendly. Okay, if you got oven cleaner on your hand, you'd get a chemical burn from it. Okay, in the same way that you would if you spilled acid on your hand. Okay, a strong base and a strong acid can be equally corrosive or caustic. All right, and then obviously basic solutions have a pH of higher than 7. Right, and as we said, they'll turn the litmus indicators to the basic colors. Okay, the basic colors tend to go more of the blue. Okay, this only goes up to 12, but if you have a strong basic solution, it'll be like borderline black. Okay, it'll be so dark blue as to be basically indistinguishable from black. Okay. All right, questions on the bases? All right, let's get everybody come up and get a Chromebook. All right, so the other stuff that's in here, guys, there's a PDF, that's just the lab sheet, okay? You can look at that when you're putting the lab report together if you want, okay? And there are 3 videos, all right? One is the preamble, what I'm gonna do today, I think that's this one, okay? One talks about the analysis and conclusion, which I'll talk about after we do the lab tomorrow. And this one is the observations video. If there was one positive coming out of COVID, it was that we were forced to video all of our labs so that people could do them virtually, okay? So there's actually a video of me doing the lab. You don't have to see me, I'm like wearing the GoPro, okay? So you can see what I'm doing, okay? And you can get all of the observations that way. So it's great if you happen to miss tomorrow, or if you just want to double check your data and make sure that it's right, okay? So you can check it that way, okay? Not a bad idea. You can kind of scroll through it quickly if you need to. All right, so you don't have to watch those videos, but here's what a lot of people who've taken my class will tell you. When you're putting your lab report together, it's a good idea to watch these ones, because I drop tons of hints and give you like really explicit instructions on what I'm looking for and what to do. All right, so that's one of the smart things to do. You can also remember, bring it in and have me look it over before it's due. Okay, so if you're done a couple of days early, just pop in, scheduled help or any of my other help times, and say, Mr. Coderre, can go over my lab, I'll pull it up on the computer, okay, and give you some tips, okay, if necessary. Sometimes I just go, wow, that's awesome, turn it in right now. Okay, and sometimes I go, maybe you should look at question number 2. That's up to you, you can choose to do that or not. All right, so when you open up that Google Doc, okay, you will see that you can edit it. It'll have your name there at the top. Okay, this is what you're going to submit to me, okay, as your lab report. Okay, so you're just going to kind of fill in the blanks here. This is exactly that format that we talked about yesterday. It starts out with a problem, okay, then you've got your variables, your hypothesis, and then procedure. And then as you go down, you'll see that there's a chart for your observations, okay, and then the analysis is just identifying the compounds, conclusion, evaluation. Class discussion, you do not have to do. All right, that's stuff that we'll talk about when I hand back the labs. We'll talk about those questions. So they are not there for you to answer. Okay, they're there for us to go over after you've handed in the lab. So please, please, please do not try to answer the class discussion questions. That's more work than I want you to do. Okay? All right, so let's talk about, okay, the lab. So what we're doing here, okay, as our problem, because your problem is blank on your sheet there, okay, the problem we're investigating here is, can physical properties be used to identify unknown compounds? That's what we're trying to do in this lab, is determine whether or not everything has its own unique set of physical properties that would be used to identify. Okay, so what you've got to do is have some sort of blurb like that, okay, in your own words as the problem. Okay, so now that you know that that's what you got to do, I'll say a couple of different ways and then you can put it in your own words. Okay, what this lab is trying to do is see if physical properties can be used to identify compounds, or if all compounds have their own unique properties, or something along those lines. It needs to just contain something about using physical properties to identify unknowns. Okay, can that be done? Okay, and I'd say for right now, guys, as we're going through this, like maybe just make like a quick note in there. I'm probably not going to give you enough time to get the whole thing written out in perfect wording, okay? But I'll give you a little bit of time at the end of class, okay, to kind of tweak things if you want, okay? Now, for the design, okay, so if we're looking at, okay, your lab report here, that's filling in manipulated, responding, and controlled variables. There's a reminder there that you need to explain your choices. Okay? 1 manipulated variable, 1 responding variable, 3 controlled variables. Now, right now, you have no idea what those are. So, I'm going to describe the experiment here, and we're going to figure out which one's which. Okay? In this experiment, here's how this is going to go. You are going to have 11 or 12 unknown compounds. You are going to run the same battery of tests on each one. Okay? So you're going to take sample A, and you're going to run the series of tests on sample A. Then you're going to take sample B, and you're going to run the same tests on sample B. Are they going to give you the same results? Okay, so does that mean then that as the experimenter, each time I run the tests, I'm running it on a different material? Okay, does that count then as a manipulation? Am I manipulating what substance is being tested? What do you think? If every time I test a different one, am I manipulating what substance is being tested every time? Okay, so what's the manipulated variable? Somebody whispered it. It's okay, I'm holding your hand through this one. Okay, it's okay, we're gonna have the right answer here. Everybody's gonna have the right answer on this one. Okay, the manipulated variable in this experiment is the unknown substances. because they're being tested each time. If each time I test a different one. Okay, so remember that in your explanation, okay, so you're gonna say the manipulated variable is the various unknown substance, something along those lines, okay? And then in your explanation, you need to tell me how that's being manipulated, which I already said, and why. Okay? Do they all have different physical properties? Okay, would that be why we're testing them all? So hopefully you've got something like this for your manipulated variable. This is kind of what I'm expecting, okay? The manipulated variable in this experiment is the unknown substances. The unknowns, a different unknown substance will be tested each time. This is the manipulation performed by the experimenter in hopes that each one will produce different physical properties. Okay, there's a how, I changed it every time. Why? Because they all have different physical properties. You don't have to have it worded exactly like that, that just has to be the gist of it. Okay, how is it being manipulated? The experimenter's testing a different one every time. Okay, why? Because they all have different physical properties. Okay, and you don't have to go about like radically changing what you had, it just has to have the gist of what I just said. Now, your responding variable is what changes, hopefully, as a result of your manipulations. So, in this lab, every time I test a new material, I should get different what? Different responses, yeah, different, in this case, our responses are physical properties. Okay? I should get, or we don't want to use I in our description here, but okay, each time a new material is tested, different physical properties should be observed. Okay, that our responding variable here is the different results of the tests. Okay, so we've said that it is the results of all of the tests. The results of the tests are physical properties, and all materials have different physical properties. We've said that now in two of them, and that's okay. Okay? In one, it was the why we were manipulating. In the second one, it's why they're responding. Okay? And that's okay. They're not always going to be exactly the same, but in this case, it kind of is. Okay? You need to tell me 3 controlled variables in this experiment. Now, before we can get to the controlled variables, we probably have to talk about the procedure a little bit here. Okay? In this experiment, as we said before, you're going to take an unknown compound and you're going to run some tests on it. Okay? The tests are observable properties. Basically, you're going to look at it and record anything that you can just readily observe about it. You don't need to write anything down right now. Okay? So you're just going to be observing everything you can about it. That would be things like color, texture, state, odor, okay? Anything else that you can determine just by being in its presence, okay? Except taste. Once you've done that, then what you'll do is a solubility test. So you'll add some water to that material, okay? And you'll see if it dissolves. Okay. Once you've done the solubility test, you will test the solution for its conductivity. So you'll run the conductivity test. And last thing you'll test is the pH. Okay? You have to test the pH last because once you put this in your solution, basically a small chemical reaction occurs, and then we can't be certain that it's pure anymore. Okay? So the pH test is always done last. Okay? So if those are the tests I'm going to run, there are probably a few things that need to be controlled in the lab. Controlled variables come in two categories. Conditional controlled variables. So that would be the conditions in which you perform the lab. Okay? Conditional variables can be things like temperature, atmospheric pressure, okay? Anything that is, kind of environmental. Okay? And the second kind of controlled variables are procedural controlled variables. Those are things you intentionally do the same each time, okay, or that you are intentionally making sure are done the same way. Okay? Those could be things like the amount of things, okay? How much water you would add, what devices you would use, how you would clean or reset those devices between trials. Okay? Things like that would all be procedural things. They're the things you actually do versus things that are just in the environment. Everyone kind of follow the difference there? You need to have, since you're going to have 3 controlled variables, you need to have 1 procedural and 1 environmental or 1 conditional, and then the other one can be whatever you want. It could be procedural or conditional, or but you have to have 1 of each for sure, and then the third one can be whatever makes more sense to you. Okay? But you have to have 1 of each. Now, if I'm trying to honestly compare these things, would it be a good idea for me to use at least approximately the same amount of them? Right? Like, it would hardly be fair if I used 2 crystals of one thing and a kilogram of another. Okay, it would hardly be a fair test. Okay, so I would say it's probably a good idea to control the amount of the unknown substances that are used. Okay? Remember, you've got to tell me how you're going to do that and why you're going to do that. I'll give you a little tip on the how. Okay? These, or this, is a depression plate. It's not sad. Okay? It just has dips in it that you can put stuff in. Okay? Those are depressions. We're going to say tomorrow that in general, what we want to do is just cover the bottom of each depression with the material. Okay? So, for solids, you'll use some form of lab scoop. There's a few different kinds of lab scoops. There's this kind, which is good for small crystals because you can just scoop them into that little dip. Okay. There's also the prison shank lab scoops. They never used to have this handle on them, but I don't know why they put this handle on it. It totally looks like a prison shank. I don't know. It's weird. Okay, but you can scoop with that. It'll depend. Okay, we probably won't use these because they're difficult to use. Okay, these will probably be the ones we use. And for your liquids, there will be eyedroppers. So you'll just squeeze the eyedropper. Okay, fill it up as best you can and squeeze it into the depression. Okay. When we do the solubility test, so when we're adding water to see if these things will dissolve, would it be a good idea to have some consistencies in the qualities of the water? Okay, what kind of water should we use, distilled or tap? Well, we talked a minute ago about how tap water conducts electricity, right? Well, won't that make all of my stuff conduct electricity then? So I better not use tap water, I better use distilled water. Okay, that's not one of your variables, I'm just saying that's something important that we're gonna do. Okay, but are some of the properties of that distilled water gonna be important to control? What are some things that affect solubility? If I wanna make more of something dissolve, what can I change about the water?
Temperature.
It's temperature. Yeah, I would say temperature is a good conditional controlled variable. Okay, all of our materials are sitting in the lab right now. They're gonna be all the same temperature because that's the temperature of the lab. Okay, we're not actively doing anything to control the temperature except doing it in the lab where the temperature is controlled. Okay, but it's certainly an important controlled variable because it can affect things like solubility. Okay, I wouldn't want to use hot water for one and cold water for another. Okay, so I've given you one of each. I'm gonna let you come up with a third. It doesn't have to be right now. You might not come up with it until after you do the lab tomorrow. That's okay, but I'm gonna let you come up with a third controlled variable. Let's talk about the hypothesis. So yesterday we said a hypothesis is a statement, and it contains the words if and then. The if part talks about what we think is the cause of the problem, or what we think is true about the experiment we're about to run, okay, something along those lines. I would say that the premise we're working on in this lab, if we want to use physical properties to identify materials, would be that all materials have their own unique what? If I'm going to use physical properties to identify them, every one has to have unique physical properties. I'd say that needs to be part of your if part. Okay, so in the if part, something about you need to word it so that something about materials all having unique physical properties is in there. If that's true, okay, that's the premise we're working on, that that is true. Okay, the and part briefly describes our experiment. So, if, all things have unique physical properties, and what are we doing in this lab? Well, various tests are being run to discover the physical properties of unknown materials. That's what we're doing. You can put that in your own words in whatever way you want, but what we're effectively doing as a quick summary is running some tests to determine the physical properties of unknown compounds. Okay, so if all things have unique physical properties and tests are run to discover these properties, okay, it's going to be more detailed than that, but that's a summary. Okay, the experiment is tests being run to find these properties or discover or determine, however you want to word that, the physical properties. The then part of a hypothesis predicts your findings or what you'll be able to do with your results. One or the other, sometimes both. Okay? So, if all things have unique physical properties, tests are run to discover them, what are we going to find? What's every single unknown gonna show me? Yeah, they should all have different, yeah, or results. We don't want to use physical properties too many times, or it seems like we're repeating ourselves over and over. Okay, they should have different results. What can I do with those results? What am I hoping to do with those results? In the end, I'm trying to identify unknown compounds. Okay? So your then part needs to tell me what you think your results will be and what you can do with them. That's what we're going to do. That's what we're going to find. Okay, so for now, this is about as far as we can go, okay, in terms of having stuff written into that document, okay? The rest of it, we have to wait until we have our results, because the next part is the results, okay? We haven't done the lab yet, so we have nothing to put into the chart that you have, okay, in your Google Doc. What you have next are the materials, okay, which we'll talk about here in a minute, and then the procedure, which we'll also talk about, okay? And then tomorrow, you'll follow that procedure and be able to fill in this chart. Okay, so you'll put in observable properties. So you might say that A was a bright pink crystalline solid, and it dissolved very easily in water, and the temperature of the water went down while it dissolved. And then you could say that it was highly conductive. The conductivity was 9 out of 10, and the pH was 6. Okay, and you would just put in that data in the chart. That's incidentally not the properties of substance A. Okay, I just made that up. Everyone kind of follow what we'll be doing tomorrow? Okay. All right, so let's talk about the materials here next. Okay, so you've got your list of materials here. Most of them are simply chemical compounds. These are the identities of the unknowns. Okay, so one of the unknowns is ammonium nitrate. One of the unknowns is copper 2 sulfate. Okay, and so on and so on. So what I would say would be a good idea, once we have our results, is for you to kind of brainstorm or research what you think the properties of each one of those substances would be. Some of them you don't have to do very much. We just found out earlier today that all copper-containing ionic compounds are going to be what? What color? Blue. Okay? And since it's ionic, they'll be blue crystalline solids. And ionic compounds generally dissolve easily in water. So I should have something that's a blue crystalline solid that dissolves easily in water, whose solution conducts or not. It's an ionic compound. Will its solution conduct or not? All ionic compounds conduct in solution. Okay, this is the process you want to go through. It'll help you figure out what each thing is. Okay, if you look up or figure out what the properties of those compounds are, then you can just match them up with your results. Okay, oh look, substance C was a blue crystalline solid that did all of these things. It must be the copper sulfate. Okay, that's the process you're going to go through. So for tomorrow's lab, okay, you'll come to the front. At the front there'll be a silver cart. On the silver cart are all of the materials that you're going to need. Okay, so you will find a whole bunch of containers of pH paper. You will find a bunch of depression plates. Okay, you will find conductivity apparatus. You'll find stirring rods. Okay, little glass stirring rods. And you will also find thermometers. You might call them a thermometer. They're the same thing. Okay, so it'll help you to measure temperature, obviously. Okay, you'll need one of those for your group. Okay, so you'll take all that back to your station, okay, and then one person will come up to the front and collect samples. Now, your depression plate can hold 6. We've got 11. I think it's 11 or 12. You have to use it twice. Okay, so what's already conveniently been done for you is somebody already wrote on the bottom of all of these the first 6 letters. right? So put substance A in there, put substance B in there, C, okay? Put them all in, just cover the bottom, take it back to your station. Now, you can perform the tests you need, okay? So you'll just observe the properties, you'll just look at them and go, okay, so A is these weird little spherical crystals and, you know, okay, doesn't really smell. Should you do the wafting test like this when you've got all of the samples in there? You'll never know which one you smell. Better to go up to the front. Send whoever is the most gifted with their sense of smell to the front. Don't send the person with a cold. I've seen it happen so many times. They send up the person who can't even breathe through their nose to do the odor test at the front. Okay? You're not going to get very good data from that person. Right, so send up the person who can still smell. If they had COVID and lost their sense of smell, don't send that person. Okay? Just have them come up and smell the samples at the front. Don't do it here. Okay? All the other ones you're going to do in here. Okay? So you'll put them in there, you put your samples in there, record just your observable properties. Color, texture, state, anything you can observe. Okay? Then you'll do your solubility test. I will give you a little squeeze bottle with distilled water in it. Okay, and you'll simply add the distilled water, let's say till it gets to be about 2/3 to 3/4 of the way full. Right, you gotta leave room to be able to stir and put in the thermometer. Okay, you wanna have the thermometer in there when you add the water or as you add the water. Because some of them are gonna change temperature immediately upon adding water. Okay, so you wanna be able to catch that. Once you've done the solubility test, so while you've got the thermometer in there, okay, so you'll have the thermometer in, another group member will add the water, okay, and you'll stir with the stirring rod. Don't stir with the thermometer. They're not strong enough, they'll break. Okay, so just put the thermometer kind of over at the edge, stir with the stirring rod. This is a quick test. Okay, I've had groups that are like stirring for 5 minutes. Look, if it doesn't dissolve after a minute, it ain't going anywhere. Okay, just move on. Okay, after a minute, okay, if it's dissolved, great. Are you gonna get them to dissolve entirely, even if they're highly soluble in this much water? Probably not. So what do you look for? What are some signs that it's actually soluble?
That it's like slowly, like you can see it getting smaller maybe?
Yeah, if it's crystals, you'll see the crystals get like rounder and shrink and things like that for sure. If the water takes on the color, if it's got a color and the water takes on the color, it's definitely dissolving. Okay, if there's a change in temperature, it's definitely dissolving. Those kind of things. So just watch for that because it's unlikely that you'll get all of it to dissolve. There might be one or two. Okay, but it's unlikely they'll get it to completely dissolve in that amount of water. So you have to kind of look. Like if it floats on the top, probably not soluble. Okay, if you can see a noticeable split within the two, not soluble. Once you've done that solubility test, then you're going to, while you're doing the solubility test, should you just go from one sample to the next like this? What should you do with this and the thermometer in between? Yeah, rinse them with what kind of water? Yeah, use the wash bottle. Okay, the little squeeze bottle I give you, that's for rinsing as well. Okay, do not rinse this with tap water, or you're going to put ions in something that shouldn't have ions in it maybe. Okay, so always rinse with distilled water. Once you've done all the solubility tests, then you do the conductivity test. So you just take the conductivity meter and you stick it in the solution and you read the number on the face. Okay, so that looks something like this. Okay, this is distilled water. It's going to read 1 or 2. Okay, that's all it reads. That's a low conductivity. Anything less than 4, we're going to say is basically molecular. Okay, if it's less than 4, it's going to be molecular. Okay, so that's what that one looks like. But if I make it suddenly a solution with an ionic compound in it, by the way, tomorrow we'll all get very zen because we'll hear a lot of clinking of glass in the lab. Okay, I mean, like that's salt. Salt's highly soluble in water, and I'm still struggling to get it all to dissolve in what is probably 10 times more water than you will use tomorrow. Okay? Okay, suddenly that's up to, well, I think it was 7 for a second there, but, okay, 7. Okay, so that's the difference between an ionic compound and a molecular compound. Pure distilled water is basically non-conductive, okay, but as soon as you put an ionic compound in it, it's gonna conduct very easily. If I had put hand sanitizer in here, which is mostly alcohol, it still would have read 1 or 2. Okay, because that's alcohol is molecular. Okay, making sense? All right. Once you've done the conductivity test, okay, then you're gonna move on to the pH test. So you'll just dump out, okay, some pieces of pH paper. I would say that 1 strip can probably do 2 compounds, so I tear it in half, okay, and just dip it in there and then compare to the color chart, because this is basically just salt water, okay? It's gonna read, well, 5, I guess, probably closest to 5 there. Yeah, probably 5. Okay, everybody all right with that? Okay, is that really, really strongly acidic? No. Anything between like 5 and 8 and 5 and 9 is basically neutral. Okay. All right, questions on how that'll work? Okay, and then once you've done that, then you just have to dispose of your chemicals. They do not go down the sink. Okay? There is a bucket at the front. I'll show you it tomorrow, but there's a big thing called the fume hood right at the front. Okay, it's got like a door on it. Okay, I'll have that lifted, and inside there's a black home hardware bucket. That is the disgust bucket, the waste bucket. All of your wastes go in there. Okay, it bubbles and fizzes like a witch's cauldron as you add things to it because it's got all kinds of nastiness inside. Okay. If you drop something in there, resist the urge to reach in to retrieve it. Okay? Or you might come out with no skin on your bones. Okay? So yeah, if you drop something in there, we'll just let it go. It'll be fine. I'll pick it up with a tongs or something. Okay? I just say that because I had a kid whose like knee-jerk reaction was to go after the thing he dropped in there, and I had to like grab him before he lost his fingers. Okay? All right, so you'll do that, you'll rinse it into the waste bucket with the wash bottle, and then you'll just take it back and clean it thoroughly before you put the next set of 5 things in it. Okay? Everybody with me there? Okay. It's pretty simple lab. Okay. Pretty simple lab for the most part. Okay. Just putting things in that dish and testing them out. Now, a few lab safety things here for tomorrow. Okay, you need to have proper footwear. Okay, and by proper footwear I mean something that covers your feet. No sandals. Okay, open-toed shoes of any kind. Okay, flip-flops or anything like that. Those are not appropriate lab footwear, because if you spill something on the table, it can run off and it'll run onto your feet. So your feet have to be covered, okay, by a proper shoe tomorrow. So bring some sneakers. Okay, that'll be fine. You don't have to wear steel-toe boots. Okay, just sneakers is fine. Just something that covers your feet. Also, appropriate footwear in terms of your being able to be mobile, all right? No stilettos, okay? Or platform shoes or anything that's, stylish but impractical. Okay, we need you to be well balanced and able to move, so please wear good, that's why sneakers are your best bet. Just bring some sneakers. In terms of clothing, okay, if you have anything that's loose that when you lean forward could get into the material, that's a no-go. All right, so if you've got like, a hoodie and the drawstrings, you got to tuck them in, tie them up, tuck them in so that they can't come down. If you got long hair, put a hair tie in your science binder so that you always have it for labs. It needs to be tied back because there's nothing worse than the smell of burnt hair because your hair ended up in hydrochloric acid. Okay, things like that. So anything that's loose is either taken off, okay, or tied up, or zipped up or fastened in some way so that it can't fall into the materials. If you wear prescription glasses, you'll still have to wear the goggles. Okay, so if you wear prescription glasses, we do have some larger goggles that will fit over top of them. Okay, if you have prescription glasses but you also have contacts, tomorrow's a good day to wear the contacts. Okay, then you can wear the nicer goggles that are lighter and easier on your face and don't leave marks. Okay, so that's up to you. Okay, but you do have to wear the goggles at all times. It's the first thing you do when you go into the lab. You go to your lab station, open the top drawer, and put the goggles on. Okay, they stay on until I tell you to take them off. Okay, and they are not forehead protectors. Okay, this is not the proper position for lab goggles. They go here. Okay, they have to be on your face, on your eyes. okay? There are lab aprons. If you want to wear one of those, you can. They're just inside the door on the right. If you want to put a lab apron on, you can. You don't have to. I would not suggest wearing your favorite clothes. Okay, maybe wear something that you're a little less attached to. Okay, or that's a little less expensive. Okay, maybe don't wear your fanciest name brand clothes tomorrow. Okay, just in case you spill something. Okay, shoes, goggles, lab aprons. Okay, behavior in the lab. Don't horse around. I have zero tolerance for that. If you're horsing around the lab, I'll kick you out. Okay, and you'll be done. You'll be doing the lab virtually with the recording. Okay, I'm not going to take people's safety. Okay, I take that very seriously. Okay, I'm not going to let you jeopardize somebody else's safety by horsing around. Okay, so just know that there's a zero tolerance on that. You don't even get a warning. I just toss you. Okay, so please be on your best behavior. I will let you pick your groups. Okay, just make sure everybody's included. Okay, if you see someone who doesn't have a group, invite them to your group. Okay, just make sure everybody is included. Maximum size of the group is going to be 4. Okay, so 4 in a group. If for some reason, by some freakish accident, you get something on you or in your eyes, okay, let me know right away. Okay, tell me what it is you got on you. Okay, and then I will take the appropriate actions for you. Most of the time it's just, if you got it on your hand, you just have to have your hand under the tap water for 5 minutes. Okay, no big deal. If you get it in your eyes, that's a bigger deal. Okay, because then I have to hold your face in the eye wash station for 5 minutes. That's not usually fun because it's usually pretty painful to have something chemical in your eyes. Okay, and I say this from experience because I once had a person get something in their eyes and then I had to put them in the eye wash station and we were both bleeding by the end of it. Okay, that wasn't fun, right? Because they get something in their eyes and they're flailing around like crazy, freaking out, and I come over to help them and they smack me right in the face, okay, but by accident, and so I'm leaking and then I'm choked because I'm leaking, and then I'm grab a handful of hair, jam their face into the eye wash station, but I did a little hard and we got them right across the nose with the eye wash bar. Yeah, but in the end, they kept their sight and, we made up after. It was all fine. But I don't want to have to do that. It's not fun. So wear the goggles, okay? Yeah, that's the best way to do it. I think that's it. No taste. We did mention the no taste test. Yes. Okay, so those are the lab safety things. Just, again, have a hair tie. Bring a hair tie tomorrow if you got long hair so that it's out of the way. Avoid anything that can fall into the stuff tomorrow. Okay? Basically, it's common sense stuff. All right. Okay. I will give you guys a little bit of time if you want to tweak any of the stuff that you've written here, hypothesis, variables, whatever. We got 10 more minutes, so you got 10 minutes to tweak those parts of it. Okay?